I'm super excited to be here this morning today in this awesome session, in this beautiful auditorium. Wow moment for me. Thank you, Ariel and Andrew, for organizing this course. That was a really great start to some theory and stuff, and now I'm going to kind of dive into some examples. So my name is Saige Rutherford. I'm a researcher in psychiatry at the University of Michigan, where I've been for a couple of years. And today I'm going to be talking about...
You have to go at the podium because to go on with the presentation, you have to go at the podium.
Yeah, because I need to switch in between code and the talk, so... Let's see if I can stop mirroring. So today I'm going to go over this model that I've been using for brain age prediction, but I also kind of want this to be generalizable in that maybe you use different types of brain images. So in my examples, I'm going to be using structural T1 weighted images to predict age, but maybe you're interested in using a different type of brain image, like a different anatomical scan, or maybe a mean task activation map or something to predict a different phenotype. So as we're walking through this together, maybe keep in mind ideas you have for how this framework could be applied to something other than just predicting brain age. Because that's, I've sort of validated that here, but I really believe that this model could work well for other phenotypes as well. So I'm going to talk about how we've adapted CNNs to be applied to brain images, some of the modifications we made from these kind of typical architectures, and talk about how I've been working on that the past few years. And also at the end, if we have time, I'll get into some ideas for even further modifications that we could make. So just to introduce myself, this is my Twitter because that's how we know each other nowadays. So I'm an analyst in psychiatry. I've been working there for a couple of years now. I work on a lot of internal kind of clinical data sets, but then also I get to work with really large-scale open data sets like the Human Connectome Project, UK Biobank, the kind of data sets that allow us to train these deep learning models. I'm very passionate about yoga and cycling and dogs. These are my dogs, Charlie and Blue. And my background is in biomedical physics. So this whole project of brain age prediction got started about 1 year ago at the Machine Learning for Healthcare conference in 2018. And there's a link to the paper here. It was led by Pascal Sturmfeld, so I want to make sure to give him credit for the original idea. And so where he started was, he's a computer scientist with no background in neuroscience, and he was kind of starting to work with my group and look at how the neuroscience field was using CNNs and kind of recognized, hey, nobody's really making any modifications for this new application. They're kind of taking these out-of-the-box models and just applying them to brain data without really making any challenges on some of the assumptions that the models make. So in this paper, he kind of started to explore that. And that was also in collaboration with Mike Engstad in my group, Chandra Sripada, who's the main PI that I work with, Mark Petersburg, and Jenna Wiens in electrical engineering. So the first assumption that we kind of wanted to challenge was this idea of spatial invariance. So CNNs were really designed to work as image recognition tasks in images. So say you have a model that is looking to identify a dog in the image. And in that image, in this natural image, I'll call it, it's going to look for a dog anywhere in the image, whether it's here, different sizes, maybe there's multiple dogs. The idea is that it's going to kind of share the parameters across this whole image, and that's why things like data augmentation works really well. You can translate your objects across the image, and the whole point is that you're looking for the same thing no matter where it appears in the images. Now, in brain data, that idea doesn't really hold because most of the data that we're working with has been aligned to group template space. And a different location in the brain represents very different information. If somebody's cerebellum is appearing flipped upside down or in a different location, that probably represents that something is wrong with your data. So our data is all in the different, in the same space, and we want to learn different information from different parts of the images. So we kind of needed to challenge that idea. And so again, the goal of kind of the standard CNNs was to detect an object in the image, but in our case, the goal is to predict brain age. So it's not to detect an object, but to learn features about the image that are going to help us predict age. So we're looking to learn unique information from each location in the image. So the second modification that we made, I'm going to call regional segmentation, and I'll walk you through this figure. I think it'll make a little bit more sense on the next slide as well. So we start with an initial 3D brain volume, and we are using 3D convolutions in my example. Many networks will use 2D convolutions, and what that does is treat each, it only considers a single slice at a time instead of considering the whole 3D shape. So using 3D convolutions was another important modification that we did. So we start with this 3D brain volume, and we're going to segment it into 2 planes as our input. So when we make 2 splits in a 3D volume, we end up with 8 different regions, which are represented in these different colors here. So when you do 2 splits, that essentially create 8 new little input regions. Whereas a baseline doesn't do that split and shares the whole filters across the entire image. Now when you do the splits and you separate the brain into these different regions and channels, an important thing to keep in mind is that you want to have minimal overlap between these regions. So you've done the effort to separate the brain into these different regions so that you can learn unique information about that region. But if you force them to be overlapped, it kind of takes away from that. So just wanted to point that out. So this is kind of a brain example of that last figure. So we start with our 3D brain volume. We apply 2 segmentations, and that's going to get us to these 8 different regions that we'll input into our network. And I just wanted to make note that these, while they're not represented here in the figure, they're still in 3D. It's really hard to make 3D figures. And they also need to be the same dimension. So even though there might be a different amount of brain in each channel, like the actual physical size of them need to be the same because the network needs images of the same size. Otherwise, it gets a little bit more complicated. So the third kind of challenge that we made to the typical CNN architecture was to apply more filters early in the network. Typically, we'll see you'll start with a few filters right at the beginning, and later in the network, you'll have many. As you get deeper into the network, you'll have many filters. Like here in this example, they start with 64, and their final layer has 512 filters. And when you're working with a model that has very distinct classes, so go back to like the animal example, if you have a model that's looking to maybe classify different types of animals or something, the filters very early in the network might be shared across your classes. So if you're classifying animals, you might have, one of your filters might be like a circle because all animals have a head. But as you get deeper into the network, your filters are really more specific to each of your classes. But when our classes are age, there's not such unique information about the different classes that you're classifying. So we kind of just had this idea like, hey, let's flip the filters to the beginning. And that actually seemed to work really well. So this is the architecture that we're I'm going to go over today. The baseline approach is is not doing any of the segmentation. We have 4 convolutional blocks followed by a hidden layer in our output of the model. So we take in a 3D image. The output is just a single number, which will be our age prediction, but it could eventually be essentially any number. It could be a fluid intelligence prediction or something. So the baseline is what we're going to compare to show that our modifications are actually improving the model. So in our proposed approach, we do the segmentation, and we do this reverse layout where we start with 64 filters, and our final layer only has 8 filters. And this is just a representation of what a convolutional block looks like in our network. Thought I'd throw that in there. So I wanted to, before we get started with the code, kind of demonstrate a couple of data sets that this model has worked super well in. So in Pascal's original paper, he worked with the Philadelphia neurodevelopmental cohort data set. We had 724 subjects, and the age range was 8 to 21, so developmental sample. And he showed that our proposed approach, where we do the reverse filter layout of 64 filters at the beginning, 8 in the final convolutional layer, and 2 segmentations, we had a mean squared error of 3 years, a mean absolute error of 1.4, and the entire network trained in just 40 minutes. We tried a couple of other experiments. We tried just doing the segmentation only, so splitting it into those regions and not trying the reverse layer, and that was, it trained a little bit quicker, but it was very similar performance. In another experiment, I've tested this idea on the UK Biobank data, which was a much larger data set, so we went from 700 subjects up to 9,000 with a much wider age range, and impressively, our mean absolute error was only 3.2 years, and while the network took 6 hours to train instead of minutes, it was a lot more data. So this is actually still quite an impressive result that I was excited to see. So I wanted to point this out. I mentioned, doing this segmentation really helped the network, but we found that there is kind of a limit to this. The more you start to segment, you'll actually start to do worse, and we think maybe that's because you're just sort of giving too much information, or there's maybe not unique information in all the different segmentations you're doing, so that is just something to keep in mind. So, okay, a couple of details about today's code. So while I just presented data from the UK Biobank and PNC data sets, both of those data sets are not the easiest to access. Like UK Biobank took us quite a long time and a bit of money to be able to download and access, and both of them have kind of a little bit stricter data use agreements. So I wanted to use data today from the Child Mind Institute because it's a really great developmental data set that I think more people should know about, but also the data use it's much easier to gain access to that data. So if this is something that you want to play around with a bit more when you get home, this data is really easy to access. And they have structural data, there's functional data, DTI. It's a very rich data source, and there's also a lot of phenotypic data if you're interested in applying this, like I said, to something other than age prediction. And so I also, I introduced this idea of just making this random split in the brain, just right down the middle twice. Today I actually want to introduce kind of a more anatomically defined split. Like we know that there's maybe unique, there's better splits and segmentations you can do of the brain other than just splitting it in half. So today we're actually going to look at splitting a T1 into gray matter, white matter, and CSF masks, and look at how that helps this model. And also just here's a link to the data portal for CMI, and there's a wonderful scientific data paper as well. So with that, we'll get started with my Jupyter, or with my Google Colab notebook. Okay, I'm connected. Oops. Maybe if I end this. Okay. Display. trying to get it so that you can see my code. Nope, still can't. Let's see. Show mirroring options. Arrangement. Somebody want to come help me quickly? Sorry. We'll get this started. I have my displays up.
Arrangement, displays arrangement, and then check that mirror displays.
Oh, small little box. Okay, cool. Okay, perfect. So like Ariel said, this notebook link is on the Brain, the website next to my name.
Could you increase the size of the font? Command plus.
Yeah. Is that a little better? Okay, we might get a bit of text cropped off, but I think that should be okay. So I, again, have a link. All of the code that we're going to be running today is in this notebook that we're going to run through together, but there's a couple of steps that I had to take in preparing this data. So if you're interested in looking at that, I do have a link to a GitHub here where I've linked some of those scripts, because while running deep learning models seems really easy when walking through a simple notebook like this, that's often not the case, and we spend a lot of time preparing our data, or what I like to call data engineering. Doing things like making sure all of your input images are the same size, quality checking, silly things like, your files need to be consistently named in a machine-readable thing, so sometimes you might need to resample or crop your images, so I've linked a few of those scripts in my GitHub if you're interested in looking at them. So let's get started. We're going to, we'll author at Google, run anyways. Hope this works. Initializing. So I'm going to install the requirements that I need for this package. They're probably pretty similar to what Ariel just used, but I think I have a few extra. So import those. Guess there's a lot of them. I forgot to do this poll right at the beginning. Can I get a show of hands of who has ever used a CNN before in their research? Okay, cool. I've used a couple of them, but maybe about 1 year ago, I presented at ISMRM a convolutional neural network, and it was my very first time. So I was just reflecting on how far I've come in a year. Running a little bit slowly today. Okay. So I imported our packages. I'm going to use Keras, which was the same library that Ariel just walked through in his tutorial. I import a couple of neuroimaging packages here, like NiLearn, which is a really awesome library that you should check out if you're not familiar with it. It has some really great image processing tools like cropping the images. Okay, I'm going to download my phenotypic file to get the ages and the subject IDs. This is also the file that I used for splitting my data into train and test, which I've already done because that took a little while. So I'm going to read in my ages, look at the distribution. So we have 906 subjects. The mean age is 10.8. Look at a histogram of them, so a little bit more skewed toward the younger folks, but it does go up to about 21 years old. I'm going to download the training and test data really quick, unzip it. Ooh, yay, this is going much quicker. Make sure it's all there, yep. Okay, so I mentioned I had already sort of converted this into our train test split. I used scikit-learn to do that, and I've already put the data into train and test NumPy arrays, which is how Keras likes to read it. Not, but like I mentioned in that GitHub repo, I have scripts that will take nifty images and convert them into this exact format of NumPy arrays if you're interested. So get my labels. I'm going to check the input shape. So the first shape is our batch, or our number of subjects that are in the training. Then we will look at essentially the XYZ dimensions, and then the fourth channel is the number of segmentations. So we're using white matter, gray matter, and CSF, so we have 3 channels there. And this is important because you need to specify in your model when you're building it, as I'll show you down here, you need to specify this variable input shape to the very first layer. The other layers will kind of learn from this first layer, but this is a very important thing to specify. So if you were using this with a different size data, you would need to change this to match your data. So won't go over all these layers too much because it was kind of what I showed you in the slides of convolution, 3D convolutions, max pooling, and I do this model summary, which shows us essentially all the information about the model that I just created, the number of parameters there. It's kind of a cool, useful thing. So I'm going to save out some of the checkpoints as we're training this, just a bunch of code to do that. I've set the validation loss here. I've set early stopping. So what this means is if our network is not getting any better in the validation set, which I don't think I defined that. So we have our train set, our validation set, and our test set. The test set is held out and not used whatsoever during training, so it's a truly out-of-sample test set. The validation set is kind of a test set, but it's used, the model looks at it when it's training, and it's kind of tells the model if you're improving. So through every number of steps, it makes a prediction in the test or in the validation set. And this early stopping value here says that, hey, if in this many of iterations through, if we're not getting better in our validation set, or maybe we're even getting worse because we've overfit our model, this says stop. I think here I've set it to 10 epochs. So if after 10 runs through all of our data, if we haven't gotten any better performance, you might as well stop because you're wasting computing power, and you might actually be hurting your performance. Some of these values here are kind of just saving some of like our outputs. Did I run that cell? No. So then I'm going to compile my model. I'm using mean squared error as the loss function. I'm also saving out mean absolute error. If you were doing something like a classification, maybe you would also want to add in accuracy here, but because this is more of a regression, a continuous thing, age here, I'm not going to use that. So I compile it, and the next step is to actually train. So this cell, I had to use this when I was training on my server because we have multiple GPUs, but it's just a little line of code that will show what GPU you're using. Okay, so here I've set the number of epochs and the batch size. This is maybe a little bit not standard here, but for this example where I'm kind of trying to train the network quickly, I've set my number of epochs to 10 and the batch size of 20. And also I set my validation split here. So what that says is I'm going to use 10% of my training data as my validation test set. So we'll set those parameters. And that is going to run, I think it took about 8 minutes. So I'm actually going to hop back to a couple of slides while this is going, but I'm just going to watch for a moment to make sure that it's getting started and how it's going. Okay, it looks like it's running. See if I can not. Already showed you that. So I just wanted to chat a little bit about some different ideas I had for this. So today I was showing segmenting. I showed you this idea of regional segmentation, where we just kind of do this random split down the middle of the brain, and in our code we're doing a split where we use white matter, gray matter, CSF. But as we know in the field, there are numerous different parcellations and atlases. So I've really been thinking about how I think this architecture could be sort of a way of testing those, maybe for different data types, different parcellations or segmentations would work better. But also keep in mind the figure I showed you, where the more segmentations we do, sometimes we can actually start to do worse. So maybe a functional atlas that has like 260 regions or something might not be the best here. Maybe we're better to stick with anatomical atlases that have, I think this one here that I'm showing has like 7 different regions. But I think that this model could be a way of sort of testing out which ones work for different phenotype predictions. I also mentioned like the idea of lesion analyses here. Maybe if you do want to use a functional atlas that has something like 200 ROIs, well, you could test just using a portion of them and then looking at how your prediction performance changes. So I think that could also be interesting. And again, I mentioned that there could be different data input types. So this model is currently set up to work with a 3D volume, so it can't handle like a functional time series, but it could handle something like a mean functional resting state image, or a subject's task activation map, or some kind of DTI summary image. So I think all of these would be really cool ideas to try out. So I wanted to point out this paper. So we kind of, this is a paper called EfficientNet, and I'll explain it a little bit. So we've kind of been talking about like all these different parameters you can tweak, different inputs that you can have to your network, different atlases, different number of filters, different how deep your network is. And these are a lot of parameters to just start trying at tuning yourself. And this is a really exciting paper that's actually being presented. I think the conference is going on right now at the International Conference on Machine Learning, and I think the computer science field was super excited about this paper. And the idea seems really straightforward to me. I was kind of surprised, like, how did nobody think of this sooner? So the idea is that you train an initial neural network with, sort of a very simple one. Maybe you wouldn't do the regional segmentations. It might just have a couple layers deep, a very simple version, but you're inputting, your image, and you're outputting your phenotype. What this kind of network introduces then is it tunes its hyperparameters itself. So what that means is it essentially chooses how many layers you should have, how many filters you should have at each layer, how many segmentations you do. So this idea kind of takes that, let's just try out a bunch of stuff and see how it works. So I think that this network could be really useful for this model that I'm introducing you today, because it can be very computationally expensive to train, so many different networks yourself. I think just in preparing for this talk, I trained like 10 different versions of this, and while I have my own server and GPU access, like it can get a little out of control sometimes. So I just wanted to point this out, and I do have a link here to the paper and the code as well, and so happy to discuss this further. I have yet to actually try it out myself, but I was reading the paper and just thought, like, wow, this is so cool, and I think it would be really applicable to this kind of architecture. Okay, another thing that I wanted to point out was this issue. So when we're predicting age, obviously the kind of thing that we're optimizing for is to say we want to get really good at predicting age, and that's the only thing that the model is caring about. Let me minimize the mean squared error between what I'm predicting and what is the true age. So we end up kind of having this forced correlation. And I guess that's not really a problem if your only goal is to predict age, but typically, at least in our lab, we have kind of like a further thing we want to do. In our case, we're looking at after we get these predictions for age, is there a deviation analysis? So for people who the model is predicting that they look younger, or older, or something, there's these extreme deviations in their age predictions. Is there anything, interesting about them that we can learn? I think originally we wanted to show that people with younger looking brains exercise more, which sadly was not the case, at least in the UK Biobank data. But so when there's this really strong correlation between predicted and true ages, we can't do as interesting of a deviation score analysis. So I haven't totally solved this issue yet. Something I've been doing in post-processing is to sort of force our sample into these like quantiles or deciles to sort of force there to be no correlation between predicted or deviation score and age, but I think that's not an ideal solution, and I'm kind of actively thinking about how we could include this in our model. I was very excited to see that there's actually a machine learning challenge this year. I think it's related to OHBM. But it was to using structural images predict age, and I was excited to see that they're thinking about this too, and that they'll give an award if your model also has a Spearman's row of like less than 0.1. You'll have the bias reduction award. So this is definitely something that I'm actively thinking about, and I think is important to address. And then I just linked some additional resources that I've been using myself. I know that there's kind of a overwhelming amount of information out there on the internet in general, but especially with like deep learning, if you're not familiar with it, you're in neuroscience, not computer science. I actively follow the literature and even find it super hard to like keep up to date on everything. So for me, I just decided I was just going to start looking at a bunch of people's code, and I didn't set out to be like, I'm going to learn deep learning and just master it. Like I just started looking at a few examples where people were using it in their research and just following along, even if I didn't fully understand everything that they were doing. I really have found the most helpful thing just to start reading people's code and seeing how other people were building their models and such. So I just thought I would point that out. Okay, now let's go back to our model, and hopefully it has finished training. Okay, so we got through 10 epochs. Was that how much? Okay, it did finish. So let's look at our performance. So after we fit the model, the next step naturally is to test it on our test set. I think our test set in this case was like 20% of the data, so it's maybe like 160 or so subjects. 182. Okay, and so this time through our these two values, the first one, 28, is our mean squared error. The second value is the mean absolute error. So this actually seems to do not that great in this case. I mentioned in the other data sets our mean error was around like 2 years. One reason might be that we didn't get through very many epochs. I think in the other data sets I got through at least like 30 or 40 before it stopped training. Also, I'll mention that the CMI data set is multi-site, which can be an issue. Maybe there's some scanner invariances and there wasn't like balances across the training and test set. I don't think I considered that when doing the split, so that could be a reason why. But so I think that trained in about like, 8 minutes or so, which was pretty fast and cool. But if you are running this yourself, I would recommend increasing this number of epochs that you're getting through, and I think that would increase your performance. So I just want to go through how to save out the model, because that's an important step after you've spent the resources to train this model. You don't want to just lose it. You want to save it and maybe be able to use it later. So this is a line that will just save it out. It will save, I've sort of listed, it saves the whole architecture of the model. It saves the actual weights. It saves the state of the optimizer. And I think I had a line up here that says, let's find it. So I'm saving like the best weights of the model. It essentially saves like when the model stopped doing the best. So even though we might have gone like a few more epochs and where we stopped increasing on our validation set, the model will save wherever it did the best. So that's important. But you do need to specify that when you're designing the model. And then also I wanted to show you guys how you can load in a pre-trained model. I've shared a couple of models that I saved when I was doing this. So we need to import another library. I saved a couple of other models where I had different batch and epoch sizes. I think this one, and maybe we can see how we perform on those. And if we do any better than 4 years. So load those guys in. So one of them I had batch size 10, epochs 20. One was batch size 5, 30 epochs. And here these go through the summary, but it's the same exact model, so I don't feel the need to print it again. You do, once you load in a saved model, you need to compile it again. So this is a kind of a repeat of the line that we had above from right before we trained, but then we're just going to use the same evaluation function, and it is the same test set that we just test the model that we trained together. So, okay, so this mean absolute error is 2.9 years, or almost 3 years, but a little bit better. I think other one was 4.9. And this was 3.1. So increasing the number of times you go over the data set increases your performance as expected. Okay, and I already finished my slides. So with that, I just want to say thank you again to everybody, to the organizers, and I'm happy to take any questions that you guys have about what we just ran through.
Thanks, Saige. I've got multimodal data, I've got MRI and PET, and I'd like to use them together. And it seems that's something similar to what you did with the grey matter, white matter CSF. Just so I got it clear, did you just combine them into a single 4D image? Cool.
Yeah, as long as they are they're the same image dimensions in 3D, this architecture could handle that. And I would be curious to see how different modalities performed.
And then if I have, if I want to enter other information that isn't an image, so in my case it would be whether it's a left or right epilepsy, could you put that into your convolutional neural networks?
Yeah, do you mean like, kind of the idea of regional segmentation where we just did that split and you would want to have the right and left like as separate, or is it not an image information?
It's not image information.
That definitely can be done, not in the current model as we just walked through. Like, are you trying to predict left or right, or?
No, I'm trying to predict a surgical outcome.
Okay.
And left or right is a predictor variable, but not imaging data.
Yeah, I mean, you could test like just maybe only giving the network the left hemisphere at a time and seeing how that works, and have a separate network to only use the right, and then see how the classification, maybe you can classify only using the left hemisphere, or maybe the right hemisphere does, but it would sort of take doing a segmentation and then testing only giving the network the left information and then only giving it the right.
Okay.
Does that make sense? I'm happy to talk more offline if you have
that'd be awesome.
questions.
Thank you.
You're welcome.
Okay, for such a question as estimating age from image data, you worked with MNI normalized data, but I imagine there's a lot of information also in the non-normalized data. Would it make sense to include them in such a model?
Absolutely. I think that even not normalized data, the data is still in close enough space that the idea that I brought up of spatial invariance still really holds. And as long as you're all of your images going into the network, even if they're not like aligned to template space, as long as they were the same size again, that could definitely still work. Or even like free surfer data, a different template or something could definitely be used here.
Thanks.
Thank you for that talk. I recently began using machine learning in the similar context of predicting brain age. And using simpler models such as penalized regression, what I find is some of the top predictors are actually quality control metrics, even in data where you exclude some of the worst scans. So I wonder, I don't know much about deep learning, but how can you know if that is the case with deep learning models, and what can you do about that?
So I think it's definitely important to know your data going in, and one of the negatives about these models, or one of the criticisms, is often that we don't know maybe what information they're using to make their prediction. So like it could be some kind of noise feature or something. So I think it's very important to, there's still not a replacement for visually looking at your data going in. And if you have poor quality data that's going into the model, that's not going to help you at all, and it's not going to help you learn anything new about the brain. So I still highly recommend that you look at all of your data before and make sure that you have high quality train and test, because that could also, like if your data in your training set is all super high quality, and then somehow in your testing set there's low quality data, that would you wouldn't do very well in the test set. So.
Thank you for the talk, and I think I just missed your reasoning behind the segmentation, what you talked about first, so could you just really shortly reiterate on it?
So with segmenting, if you don't do that segmentation, the network will share the same filters across the entire image. And so the segmentation forces the network to learn more specific information for each region, because we believe, like in the example I did with the dog, we're trying to essentially find the same object across the entire image, whereas in the brain we know there's really unique information in each region, and so we want to kind of force the network to focus on that.
Thank you.